Where was it?
Good afternoon. You're watching LNA Now with Barry and Jenny. I am Jenny Nagel.
And I'm Barry Agnew. Happy Wednesday. And I'm safely going to say 3/4 through summer school. Today is July 23rd.
Third.
And I just, on Sunday, when I woke up, I looked at the calendar and saw it was July 20th. And I said, we're going to blink and it's going to be Labor Day. Because like that last few weeks of July just like disappears. So yes, we are 3/4 of the way through summer school. And today, you guys are in for it. You're in for some major education and some major science. So I hope you have your pens and your paper ready. Jenny, are your pens and papers ready? I know mine is because I was already asking questions in the green room.
And you guys, if you can think back, if you've been part of the LNA family for a while, we've had Factor 5 on a webinar of LNA Now prior. And the last time Barry and I were sitting like this, thank goodness our cameras were off because we were just like gawking into the screen because it was really when growth factor stem cells, it was still fairly new, right? It was new conversation and new education. And I feel like today, while exosomes have been hot topics for a while now, this is really an opportunity to dive into the sectors and how they fit together and really get some education. And you guys will, because you're joining us live, you will get to ask the questions that are all over the internet right now. There's misconceptions, and I know we've got the team here that can help demystify this.
Who's here? We haven't said anything yet. Maybe we should introduce them. Can we introduce them? Here we
I
go.
suppose.
Today, we have the entire, well, not the entire, we have a large portion of the education people from Factor 5 here. And number one, and most notably, is John, who's the CEO and founder of Factor 5. And I'm going to let John take over to introduce the rest of his team. But quickly, I just wanted to say, John, before you get started, like this is a controversial hot topic in the industry. One of the other brands I know that's getting into exosomes sent us a TikTok of this guy claiming to have a PhD, just blasting exosomes. And I sent the other this guy back a message that said, I'm not quite sure. The guy, first of all, the guy looked, I don't know how he has a PhD. He looked 12. And I said, I don't necessarily understand how multinational corporations can be selling these products globally and have some 12-year-olds say that they're ineffective. I think that's really like just a TikTok scam to try to get views. So had our own Dr. Wong. He just submitted his editorial article on exosomes, and he highlighted 4 kinds of exosomes: plant, yeast, human, and synthetic. And he had sort of like a little bit of feedback on each of them. And I know you center on a specific one that I think can have a lot of, shall we say, opinions on. And so I'm really excited to hear how you guys bring this specific exosome into cosmeceuticals in a safe and effective manner. So I'm going to let you take over and I'm going to let you introduce your team. And I'd like everybody that's being introduced, I want like 2 interesting things about yourself, because the community here at Lipgloss and Aftershave really likes to connect. They really like to get to know the people that are presenting, and it forms a relationship that I think makes our interactions really successful. So John, take it away.
Well, thank you. What an introduction that was. So thank you, Barry and Jenny. It's a pleasure to be back with you. Thank you for all of you that have tuned in. We find it really gratifying that anybody would care, what we think. So it's always great when we get people to come and bring your questions. I want to thank my co-presenters today, Dr. David Statura, here with me. I'm going to share 2 interesting things about him, and the lovely Tiffany Underwood, who is in the other screen. So I agree with you. I want to just start out with this. Let's get it all out on the table. Exosomes are a controversial hot topic. Factor 5, we've been in the business of stem cells for a long time. And so we felt so much that it was a hot topic that we even know we've been deriving exosomes forever. but it's not something we really wanted to weigh in on until we felt ready to do so, or we felt the market was ready for us to do so. And so for us to do that, I wanted you guys to have the best education possible. So I brought two of the best educators that I know. The first is my good friend and colleague, Dr. David Statura. He is the Chief Science Officer. He looks like a floating head right now. He's the Chief Science... He is the Chief Science Officer here at FACTORFIVE. So whenever I say anything that sounds relatively intelligent, I'm basically cribbing all my notes from this guy over here. He is an Ivy League trained molecular biologist and immunologist from University of Pennsylvania. He's a stem cell scientist, researcher, multiple publications in the field of hematology. He's a very, very smart guy. And a couple of interesting things about Dr. Dave. He wins best costume in our Halloween pageant every single year. So he's a fantastic costume designer, apart from being, and he's the funniest PhD that I know. So he's got a great sense of humor. Those are two interesting things about him. Tiffany Underwood, the two of you know, I think your audience might know her a little bit as well.
Yeah, they know Miss Tiffany.
Yeah, licensed esthetician. She has an undergraduate degree in biology. She knows a lot about the chemistry, sort of birds of a feather. When we first met her, we felt a kinship with her. We loved the charm, the Southern accent, but we just felt like she was somebody who the appreciation that we feel in our heart about our customers, they're small business owners, a lot of them, they're taking risks buying our products. We feel an appreciation for them that is hard to relate. It's hard to relate when other people don't feel that. But when we met Tiffany, that was the thing that came shining through from her, is that she loves them as much as we do. So a couple interesting things about Tiff. I don't know. You guys already know Tiffany, but she's from a small town in Georgia. And she invites us to go fishing on the coast with her all the time, which we were definitely going to take her up on. And
she invited me to the coast, but not to fishing. She wants to take me shopping in Savannah, but she knows her audience there. So that's, it's to her credit.
Well, one interesting thing about me is I still like to train Brazilian Jiu-Jitsu, and I got punched in the face this morning. So please ignore this. I feel self-conscious about it, but it was an elbow to the jaw. I'm fine.
I think you've got some
that's
healing
amazing.
products. I think you have some healing products at your disposal, John.
Yes,
I'd be willing to guess.
Yeah.
But John, not to take over, not to like co-opt your introduction, but you really you said that anything that you learned about or anything you talk about when it comes to science comes from Dave. And I have a question, like, what are the letters behind your name, sir?
Mine? So
John. I know.
Yeah.
John, I think you have a couple of letters behind it that should give you some street cred when it comes to talking about science. Why don't you talk about that?
I have a doctorate. I'm a PhD, but my PhD is I'm more in the field of economics and business. So I do have a PhD, and I have studied a lot of things. And my research is on pharmaceutical research efficiency. So I've been in the field of drug development my whole career. It's something I've always studied. But I just want to pass along to you and my audience that Dr. David Statura is just a fantastic educator. And that's really what I'm trying to convey, is that he's always been able to teach me things in a way that is really easy for me to understand it.
Yeah, I've read his bio when he teaches at IECSC, and he's always someone that we see in passing waving as he's walking to a class or standing there talking to a group of people at the show, just giving them all the information. So we're really excited to be able to have this class today. I didn't know you were Brazilian Jiu-Jitsu. Isn't it, that's Israeli. There's an Israeli martial art that has like two words to it.
Krav Maga.
Yeah, I tried one class of that and I walked out about 20 minutes into it. I am not designed for those types of extracurricular activities. This is not my thing.
They say that jiu-jitsu is a thinking man's game, so I've always enjoyed it. It's like playing chess with the human body. So it's really fun martial art.
That's amazing. So we want to just do a real quick recap. For those of you it takes a while for everybody to like realize, oh, it's 2 o'clock, I got to log on. So we've doubled our audience size watching since we've started. Welcome to summer school. It's Wednesday, July 23rd. We're about 3/4 of the way through. And today we are here with a team from FACTORFIVE to talk about their all new exosome treatment product. We've got John, Dave, and Tiffany presenting today. John is the CEO, Dave's Chief Science Officer, and Tiffany is a biochemist and esthetician. So if you don't learn something today, you're not paying attention. Couple of housekeeping tips. The first thing that we like to do is a little bit of a roll call. So John, we've done this in the past. We just started it in summer school and people are already in the chat. Jenny, are you seeing the chat? They're already in the chat telling us who they are and where they're from because they've been here. So if you go into the chat, drop your name and where you're from so we know where people are watching from. So far, we've got Mary Ellen from New Jersey, we've got Alicia from Texas, and we've got Sue from New York. Angela's coming in from Canada. We've got Edward from Texas. Oh, here we go, Seattle. Pencil, I can't keep up. You guys are killing it. I love it. The point is that is the chat. So that's where you have fun, you chat, you tell people you like something. It is not where you use, or it's not where you go to ask a question. We are imploring you to use the Q&A because we can download the Q&A and then we can send that over to FACTORFIVE. And that will give them your name, your email address, and your question. And so if we're not able to really thoroughly answer your question or even get to your question, the team at FACTORFIVE will be able to get back with you and answer your question specifically. And John, just so you know, it's heavily engaged over here. We are getting emails 10 minutes after the webinar is closed. When do I get the replay? And it's like, there's a process. And so people are engaged. So there's going to be questions. You can already see the chats exploding. And we got a lot to cover. So I'm going to let you take over. Jenny, do you want to work with him on the sharing of the screen concept?
Yep, John, you are ready to go. And you guys, just so you know that we've got Caitlin also here from FACTORFIVE. She is in the background ready to help answer your questions. So don't fear, they will be answered in a timely manner. John, you are ready to go. We can see your screen and we will go off camera.
Terrific. Everybody can see it then? All right.
Yes.
Allow. Okay. Well, thank you again. As I mentioned before, this is a controversial topic, and so it's not something we wanted to dive right into at FACTORFIVE. In fact, we really became inspired to get involved with exosomes only after we had a lot of our customers in the marketplace asking us questions about it. So for us, it's very gratifying that you guys trust us enough to jump on and to hear what we think about it. So welcome. We've kind of already done this. I just want you once again to know how much we appreciate you, our customers, and the people that we are educating. So who is FACTORFIVE? We're a biotech skincare company out of Northern California. Dr. Dave and I are sitting in our manufacturing facilities here in Chico, California. We culture our own human stem cells here. We actually mix the formulations ourselves, put them into bottles, and put them in. So we do everything in-house, vertically integrated. For us, that was just a function of wanting to do right by you, our customer. Whenever you outsource production of a product, especially one as highly technical and scientific as ours, you risk losing the essence of the beautiful things that we're creating, right? The proteins and the stem cell growth factors, the exosomes. And so we do manufacturing ourselves in-house. So why exosomes and why now? I sort of touched on this at the beginning, but this is it's a controversial topic. Why now? Why exosomes? First, why exosomes? Exosomes are a derivative of human stem cells, and they are something that human stem cells produce and generate. So as a human stem cell biotech company of some tenure, we've been working with exosomes literally for over a decade. And so a lot of people are sort of wondering, like, why didn't you come out with an exosome product? Or why aren't you taking the lead in the marketplace about talking about exosomes? Why are you guys quiet when you got a bunch of molecular biologists and chemists and stuff on staff who can train on exosomes, you know? And our answer is because a lot of what we were seeing in the marketplace in terms of the claims that were being made, we didn't feel were accurate scientifically. We didn't feel like it was a good representation of the science. And so it just sort of felt like a weird space we didn't want to get into. Why? For some of the reasons you see on your screen. Exosomes are extremely temperature sensitive. So they need to be kept extremely cold if you're going to be working with them in a normal environment. They got to be kept cold, like really cold, like minus 80 degrees Celsius. So we were seeing people claiming that our exosomes are shelf stable in this liquid serum that's on the shelf. And we were like, well, that's bullshit. You know, like we knew right away that there were problems with what was being said on the marketplace. Exosomes are also exceptionally hard to isolate. Like I was saying, they come from stem cells, but after you do the cell culture, which in and of itself is very, very difficult, you have to have secondary processes to separate out the very, very, very small exosomes from the other particles that a stem cell expresses. So it's not just child's play to be able to isolate and use exosomes. And we're hearing these people, they have like law degrees, they have some background in engineering, and they're like, I got the best exosome product. And we're like, that just doesn't feel accurate. They're hard to keep stable. So another thing that we've seen are products that have, well, they have no way of keeping. Exosomes are defined in some way. I'm going to let Dr. Dave teach you about this, but they're defined in some way by the barrier that they have around them. They have like a cell membrane around them. And it's hard to keep that cell membrane stable. And they're exceptionally hard to detect and test. Even the best labs in the world, which we're going to go into a little bit. We don't want to overwhelm you, but even the best labs in the world are having a hard time determining what is an exosome versus what is some other extracellular particle. So the exosomes are extremely complex. That's kind of what I'm trying to get at. Now, I think, is this the next slide? Okay, wait. So I'm not going to go forward. But in contrast to what the man you were telling us about, the TikTok PhD, was saying, they are widely studied right now because they're a fascinating biological particle that we're learning more and more about it. But in virtually every study, there's tons of publications out there. Anybody can read them. We encourage you all to do so. Go on Google Scholar, go on PubMed, type in exosomes. In virtually every publication, they're having incredible outsized benefits for what they actually are, what they do. So it is a medicine of the future that's being widely studied right now, but it's also very, very difficult. I hope that both of those things stay in balance in your mind. Okay. Is this one mine or yours?
This one's mine.
There you go.
Hi, everybody. So I'm Dr. Dave. I'm the Chief Scientific Officer, if you missed that, over here at FACTORFIVE. So John and I work together to kind of plan and plot the trajectory of what we're working on. And John wanted me to speak a little bit about exosomes and what they are, how important they are, how we detect them, how we package them, those kind of things. And so I figured I'd kind of just start off with, what is an exosome? And I also wanted to mention, Barry and John have brought up, this TikTok thing with somebody who's 12 years old. I'm clearly not 12 years old, right? I am a wizened
he's a science wizard.
Science person who's been in the field for a long time. Most of my graduate and postgraduate work has been on adult stem cells. So I do a lot of this work on a daily basis, and I have for like 25 years. So I think the best and easiest definition of an EV or an exosome is basically they're these small vesicles that are secreted from cells, and they're about 30 to 150 nanometers in size. And just as a comparison, that is super, super tiny. So a red blood cell is about 8,000 nanometers in size. So basically a red blood cell is about 250 times larger than these little tiny things that you're seeing on the screen. And we have taken, so the images that we're showing you right here, these are actually electron microscopy, okay? So you cannot see these things with a normal microscope. They're far too small. You basically need to take them, put them on a slide, cover them with something that reflects electrons, and then hit them with an electron beam. And these are the images that you get out. And so on the left, I'm just gonna, if you look at that white arrow, I'm not sure if you can see our cursor, so I'm not gonna bother with it, but that white arrow is showing that is an exosome, okay? We'll talk a little bit about some of the nomenclature of exosomes versus extracellular vesicles in a minute. but that's what they look like. They just look like something wrapped in like John mentioned, a cell membrane. So you can see that line, and what's inside of it from this image is a mystery. Okay, we'll talk a little bit about that too. And what I want to also point out is that on the right side, there are two blue arrows. And hopefully you can see, hopefully it comes out on your screen, that those are extracellular vesicles or exosomes. But if you look carefully, you can see that there's one inside of another inside of another. So there's multiple layers of the cell membrane that is inside of that extracellular vesicle, okay? And so these are, people don't know why this happens. We know how it happens, but these are things that are being currently investigated by scientists, you know, in our laboratory, but also in research institutions around the world. And so let's just do a summary. So exosomes, they are called extracellular vesicles. They're generated by all cells. What's inside of those things? It can be RNA, ribonucleic acid. It could be DNA, deoxyribonucleic acid. It can be proteins, could be lipids, and it can also be metabolites. So when I say that, it's just basically breakdown products from metabolic activities that all cells are doing. These things can mediate signals between cells, short distance and long distance. They're important in health and disease, and they affect various processes in cell biology. We'll talk a little bit more about that. And I'm sure, again, there's going to be tons of questions. We're happy to ask, you know, we're happy to answer them. So if you're thinking about stuff and I'm saying something that triggers something, you know, throw it in the Q&A and we'll get to it later. I want to mention that I think in the literature, everybody says, you know, extracellular, and I'm going to use the terms interchangeably. Okay, so I'm going to use the term extracellular vesicle and exosomes interchangeably. And the reason that I do that is all scientific versions of this, people refer to these as extracellular vesicles. Okay, so in the scientific literature, we call them EVs or extracellular vesicles. We don't necessarily call them exosomes. I like you guys to think about exosomes are like a square and EVs are like a rectangle. So all exosomes are rectangles, right? All squares are rectangles, but not all rectangles are squares. Okay, so EV is the term that kind of encompasses anything that's secreted by the cell that is wrapped in a lipid membrane. And so the reason that we call them EVs versus exosomes is the definition of an exosome is something that is produced through a very specific pathway inside of the cell called the endocytic pathway. And if it's not produced by the endocytic pathway, then it's not technically an exosome. And it's one of these things where you need to visualize that actually in real time, and it's impossible to do so. So basically scientists call them all EVs. John is thinking of an amusing anecdote that he will share with you in a minute about EVs. But I think that the standardized definition that most scientists recognize is that they are these particles that are secreted from cells. They're about 30 to 150 nanometers, like I mentioned, and they have specific markers on their surface. So you see those membranes that are there. Those membranes have these things called tetraspanins, which you can think of as a protein that spans those that membrane bilayer. And you can attach an antibody with a color onto it and actually detect what are the markers that are actually on those membranes. And so there are a couple markers. We use 3 specific markers called CD9, CD81, and CD63. So if they have those on the surface, they're very likely an EV. And then we also do cryo electron microscopy, which is what we're showing you here with these images. So the image of basically a fixed, and I'm going to say the word dead, but of course they're not really alive because they don't have a nucleus. And we also use nanoparticle tracking. And for those of you who see a lot of the quantitation that comes out in most of the aesthetics world, most people are just basing that on nanoparticle tracking or NTA. And basically it's just the machine that is looking at very small particles. But as you can imagine, there's no specificity to what those particles are. And I'll also tell you that the range of limitation, the range of that machine is basically 70 microns and up. And if you're paying attention, I just told you that they're 30 to 150 nanometers. And so basically between 30 and 70, you're missing, right? So a lot of these smaller EVs or exosomes, you're actually not even quantitating with that NTA analysis. And I also want to point out that, because of this heterogeneity, we don't really know exactly what's inside of each one of these exosomes. We'll talk a little bit later about that. Yeah, we're doing research to try to figure out specifically what is inside of these exosomes.
Yeah. So a couple of things I just want to add just from these pictures. These pictures are actually pictures of our work. So these exosomes are derived from our labs in Northern California. Sometimes you see people, they're downloading pictures from, like I said, PubMed or Google Scholars. So these are our exosomes. We're really excited about it. And one thing that you guys will find out if you're really, really excited or interested in this topic is that exosomes are largely being studied because of their ability to attach to other cells very fluidly. They can carry a package, if you will, from one cell to the other in a way that is really, really difficult for anybody in pharmacology to mimic. So if you can imagine some big drug company, they really want their drug molecule to get into your cells so it can work on your cells and affect a change. It's very hard to do that. We can go into all the reasons why, but it's just really, really hard to get a drug molecule to deliver into the cells. With an exosome, it's extremely easy to do that. Exosomes deliver packages to cells really, really well. So that's one of the reasons why they're so exciting and being studied today.
Yeah, and I guess, kind of piggybacking off of that with John is, all of us are probably aware of the COVID vaccines that came out during the pandemic of 2019. And you could think of the COVID vaccine as a functional exosome, right? So the COVID vaccine is literally an mRNA molecule, and it is wrapped in a lipid. And basically that lipid fuses to your cells and basically deposits the mRNA inside of the cell. So you can think about the fact that exosomes, I mean, obviously that's not a natural exosome, right? That is a synthetic exosome. But the concept here is the same, where you're taking a signal and you're putting it into a lipid layer, and that will fuse very well with the cell and deposit that signal inside of the cell. So
it's really concept. Yeah, it's really, really like the cutting edge future of drug delivery, which again, I don't want to make this too complicated for everybody, but this could have implications on literally every single disease that we know. Eventually I believe that it will, because it's such a more effective way for us to get the medicine into our cells. So you can imagine it as a message delivery system. We have a natural one, a biological one that your cells are doing right now all the time, and that companies like ours are studying and sort of trying to leverage. But it also can be a synthetic delivery system for pharmacology and drug delivery in the future. I hope that sort of helps to summarize the overall subject of what an exosome is. But how do we get our exosomes, Dr.
Yeah, how do we get them? So I'm going to give you a brief explanation of where we get our exosomes. And I think this kind of really highlights a lot of the issues in the field, because really what you want an exosome. We're going to talk a little bit about how exosomes signal to other cells, but really you want an exosome that would normally be signaling to the cells that you want to either cause regeneration, repair, anything. So what we do is we use adipose-derived mesenchymal stem cells, ADMSCs. That's a mouthful, but basically these are the stem cells that reside in the hypodermis of the skin, and they give rise to basically all your skin. So for those of you who don't know, if you didn't have stem cells, if you didn't have adult stem cells, you would be dead, right? So a lot of our tissue is constantly replenishing. One of those things is the skin. And one of the stem cells, the main stem cell population that replenishes the skin, are these adipose-derived mesenchymal stem cells. So on a daily basis, they are cranking out millions of cells to replace the other skin cells and moving away from the basal layer out to the surface of your body, replacing those dead cells. So these are the right kind of stem cells to be signaling to your skin, which is constantly being regenerated, but also think about the fact that it's constantly, under assault, right? So UV damage is bad for your skin. Microneedling is bad for your skin. All of these things that we do in skincare, it's not bad for your skin.
You gotta tell the audience.
Yeah.
Microneedling causes surface layer trauma to your skin. That's what he's saying. It's good when it heals, but it does, there is sort of there's a yin and a yang to that.
Sure,
Proceed.
sure. Yes, thank you, John. So what these stem cells do is they send out signals to basically repair the trauma, okay, whether it be minor or major. And I'll tell you over here, it's kind of a slightly busy slide, but let me just show you how we get them. We basically find people who want to undergo liposuction, and they have to be healthy and they have to be young. So we screen them. They have to be between 18 and 24 years old, no prior medical conditions, no inflammatory issues. And then we get consent to take that liposuction tissue. And as you can see in that top beaker there, there's this layer of adipose, that's just the fat. And then there is this layer of blue. The cells aren't actually blue, but this is a pretty color that matches all of our color schemes. So we isolate those cells out of there. We get rid of the adipose tissue and we grow them. And this is the really important thing, and John's going to talk about this a little bit later. We grow them in a way that simulates that there is a wound, an active wound inside of the body. And so what do those stem cells do? Those stem cells want to secrete proteins, and they want to secrete exosomes that are going to help repair that damage that the cells think is occurring inside of the body. And then what we do is we collect those growth factors, the proteins that are being secreted, extracellular matrix proteins, everything these cells are cranking out, especially these exosomes. And then we take those and we basically process them and make an exosome product. The exosome product that we make also has the growth factors incorporated, because I want to stress to you that the cells don't just talk to each other with exosomes, right? That is not the main way that cells communicate with other cells. The main way that cells communicate with other cells is through proteins that they secrete. I have a couple of videos that I'll show you in a couple slides, but the exosomes are an important part of that process, but they're not the only part of that process. And so I think that's a really important thing to think about when you're, kind of talking about this in the aesthetic realm.
Can I add something?
Yeah, sure, of course.
Here's one of my favorite fun facts that I hope you guys can easily attach yourself to. The human body has stem cells all over, but the tissue type in the human body that has the most stem cells might surprise you. Some people might think of bone marrow, or you might think of umbilical cord blood, or is popular. But the tissue type that has the most adult human stem cells is actually fat tissue. And some studies indicate that it has up to 1,000 times more stem cells in fat tissue than there are in other tissue sources. So it's very, very abundant with adult human stem cells. So when Dr. Dave is sort of talking about how we use fat tissue, it's not for nothing. It's not for no reason. First of all, the fat resides right next to your skin, in which he was explaining, and that's the stem cell type that's most appropriate to heal your skin or repair, replenish your skin. And secondly, they're just very, very abundant. They're easy to get to. They're ethical. Nobody's worried about us using their adipose or their liposuction, right? We're not studying embryonic stem cells, anything like that. So it's an excellent stem cell source for the kind of things that we're doing, and it's ethical, which is important to us. So it's a fun fact that I hope you guys can appreciate as well.
Yeah, thanks, John. And so these are the videos that John doesn't have access to play. But if he had access to play,
just describe what would happen. I want to hear your play-by-play.
Yeah, this is the play-by-play. So as I mentioned on the last slide, cells aren't just secreting exosomes, okay? Cells mostly talk to each other by secreting proteins out of themselves. So there's a whole pathway. It's the same pathway that exosomes are made in. The only difference is that exosomes are wrapped in a lipid bilayer, okay? So basically, as a protein is sent out into the world, into the extracellular matrix around the cell, the exosome, basically, it wraps it. So as the protein's coming out of the cell, it kind of blebs off with this cell membrane and floats away. But the main way that cells usually do this secretion is they just secrete a protein. So the cell membrane is here, the protein is in something, but it basically comes up, opens up like that, and then the protein comes out of that vesicle. This is my side job doing animations with my hands. I hope
You
everyone
do an interpretive
can
dance
appreciate
really quick. Can you
this.
hop up?
Maybe
and show
later,
us what
maybe
that would
later.
look like.
But the key here is that cells aren't just secreting exosomes. Cells are also secreting a lot of different signals that are just going off into the extracellular matrix. One of the ways that this happens is called paracrine signaling. Para means next to, like a paramedic. So paracrine means next to. There's also cells that do juxtacrine signaling. That means that the cell actually has to be physically touching each other, and there's a signal that can pass in between those different cells. So cells do a lot of different talking to each other and telling other cells what to do. And this is exactly what happens when you have a wound, right? So let's say you get a cut in your skin. The cells that are important for this signaling are those adipose-derived mesenchymal stem cells. And they're going to sense that there's an issue, and they're going to start secreting proteins that are telling immune cells to come over to fight bacteria. They're going to send out extracellular matrix proteins like collagen to start repairing that wound. They're going to send out things to clot if there's enough damage that has caused blood loss. So basically, those protein signals are mostly secreted through this paracrine signaling pathway. But on a minor level, well, we don't really know. I mean, people are still investigating this. There's also exosomes that are being secreted by those cells. And obviously, those will fuse with other cells and give them signals to do things.
Yeah, it's unfortunate we can't show you guys the images, but just to top line it, maybe you guys can remember when you're first studying aesthetics and you could hear about a product that had like now enhanced with epidermal growth factor. It was like the one cool new thing, and then sort of advanced, and we had fibroblast conditioned media, which had like 100 things, and then we had stem cell conditioned media, which has like 700 things. And now when we're adding exosome, we just keep, basically you can imagine the biological process that it takes for your body to heal itself is extremely interesting and complex. Multiple layers of different things going on all at the same time that heal your body. And all of those things need to be communicated. It doesn't just happen. It has to have some precursor, some instruction from the brain and from the cells and stuff. And so all of these little things that Dr. Dave is talking about play a role in that. Large or small, they're contributing to the normal healthy function of your cells. And the deeper we go in studying that, is sort of the more interesting but more complex it gets. We started out at that superficial layer of it. Hey, there's this one cool protein, epidermal growth factor, and now we're just getting deeper and deeper and deeper into it. So it can be overwhelming, but just to remember that all of these things contribute to the normal healthy function of the skin, which starts to diminish over time, which is why we get aging. One of the reasons you get aging and aging processes. And so molecular biologists and the smartest people you've ever heard of, they're studying this to try to understand, if you really get to the heart of how we're healing ourselves, think about it. When you get a cut, it's not the doctor that heals you. The doctor just can stitch you up. It's your body that heals itself. It's always been that way. It always will be that way. Your body heals itself. So that means somewhere deep inside your cells and deep inside your body is this knowledge, I'll call it that, of how to heal yourself, or even further is the power of how to heal the human body. That's all baked inside of your cells. So these scientists, and we're studying that to try to understand and optimize that power, to try to give you better power to heal itself. So we're extracting these things, studying them in the lab, trying to figure out what they all do to put together to make you heal. And so in the aesthetic world, there's a million applications of that type of potency and power. And so we'll talk a little bit about that, but in a nutshell, that is the totality of our discussion with you today, is what the human body is doing. It's healing itself, or it's regenerating itself, or it's cells dividing, or it's producing more collagen, or a million wonderful, healthy, normal functions. How is that happening at the cellular level? And these scientists, these researchers, they're publishing these papers, and now it's entering into our world here in aesthetic medicine. And we are trying to take all that and translate it into a way that you can understand it and apply it in your day-to-day practice. So it's a fascinating and diverse field of research. Let me see. Is this mine?
Caitlin's telling us we should turn off our, there we go. Does that work better?
Oh,
Can I
cool.
can I pop in and ask a question? So
Sure.
when we were in the green room and like at the very beginning of this, we talked about the use of human exosomes. And what what I want to find out is the we have Dr. Wong, one of our medical contributors, is based in London, and he was talking about he wrote an article for us on exosomes because he's worked with exosomes in cosmeceuticals in the past, and he talked about umbilical cord mesenchymal uh, exosomes, and he said that they are not approved for use in Europe, and I thought it was I can't UK and the EU. So obviously mesenchymal. I actually I don't know this. Jenny asked this question.
Yeah.
Umbilical cord and adipose tissue mesenchymal stem cells, are they different or are they just mesenchymal stem cells from the human body?
Mesenchyme basically refers to kind of a tissue layer that's formed during development. And
Okay.
so all of these cells, it's again, it's like a square is a rectangle, but not all rectangles are squares. So all of these cells are mesenchymal stem cells, but they like the subsets of them are slightly different. So they all come from the original same tissue type. But Barry, what you're really asking is true in that the EU does not allow basically human stem cell products. So there are multiple places throughout the world that have that regulation, but the EU is very strict about basically having things that have touched a human cell.
Okay.
They do not in their packaging. Yeah.
And,
Okay.
and and just to piggyback on that, and this is really important for our audience. What we are talking about today, we are trying to educate you about the whole field. So we're taking a big bite out of something that's very complex. But we also want you to understand we're trying to narrow it down to the field of aesthetic medicine. In the world where we all operate in aesthetic medicine, the claims that we're making are really what's regulated by the FDA. So exosomes we're teaching you the potential of what exosomes are being studied to do all across multiple disciplines of medicine, okay? But in the field of aesthetics, we're still just talking about the same basic things about reducing the appearance of wrinkles, about reducing the saggy skin by helping to calm inflammation, et cetera. And so we are advancing the subject of exosomes within our field and within the basic claims that are allowable by the FDA. We're not advocating that, hey, exosomes will, heal your skin cancer or some very, very medical claim. We're not doing that. We're teaching you that exosomes are being used and studied in the lab and by other biotech companies to try to become medicine, to try to do all of the things that heal everything. But in our field, we have some very specific hopes that you can get out of our products and out of the exosomes that we're making. But it's very narrowly focused onto your world of aesthetic medicine.
So I want to like pop on for like one more layer. So one of the things that I have seen throughout like the industry, both in many different industries, when it comes to the UK, the EU, and the US, is there are things that are allowed in the US that aren't allowed in the UK and the EU, and then there are things that are allowed in the UK and the EU that aren't allowed in the US. So if, let's say, for example, people are, somebody finds, they discover that, okay, they see on the internet, they see that kid on TikTok that exosomes don't have this, or exosomes aren't allowed for use here, or exosomes really aren't effective. What do you say to the esthetician who is considering exofecta and says, but I didn't think exosomes were allowed in the EU? Like, is that something that is, are there other things that aren't allowed in the EU that are prevalent in the US market as well?
I take that one.
Yeah,
Yeah, so this is a great question, and it's a cause of a lot of confusion out there. It's really simple. If you can, it's like two different games with two different sets of rules, right? Like in the United States, we have a regulatory body which we should all adhere to called the FDA, right? The Food and Drug
Yep.
Administration. In the EU, they have a completely different regulatory body. It doesn't mean the science changes. It just means that different governing bodies with a different agenda are setting the rules to the game. And so for the European Union, they have much more stringent policies around a few several things, in fact, some of which I totally agree with, some of which I don't agree with, but it's just government types. I mean, they're not necessarily all scientists, and they have an agenda. And so the science is the same, though. Exosomes do what they do, whether you're from France or you're from America. But so we're trying to teach you the science. But if you are somebody who says, listen, I want to do right by my clients, I know this is banned in the EU, so I'm concerned about that. I think that's a legitimate concern. You're probably wondering, like, why is it banned? Is it a bad thing? Should I be nervous about it? And the reality is the folks from the EU are missing out on some of the most exciting advancements in several fields of medicine, both in the aesthetic world and on other places, because they have such stringent regulations. Now, the flip side of that is any company, whether it's ours or anybody out there, who is willing to go through all the layers of regulatory scrutiny that the European Union requires. They're willing to do the battery of tests and more tests and more tests. And we're talking about possibly a decade of testing and many, many millions and millions of dollars. Anybody who's willing to do that can be approved for anything in the European Union. They're a regulatory body, and if you meet their requirements, you can be approved over there. The fact that
John, I love that. Thank you so much, because I knew you had an answer, because you don't take a product to an international trade show without that sort of like explanation. And I think that when people listen to this, it completely explains the situation, and I love it. Question, I know that Health Canada is like the same sort of situation as the EU. Do you guys distribute this in Canada?
Health Canada has been extremely easy to work with for us up to this point. I'm a pretty transparent guy, and so we're still in the process of working with Health Canada, but they've been extremely reasonable. You can imagine, I'm not trying to cast stones, but you can imagine that when EU says, yeah, give us a decade and $50 million worth of tests, that's a little bit of a pouring cold water on us and our science. It's hard to do. Health Canada has their own stringent testing. They have their own regulations they want us to meet, but we found them to be very reasonable and working with us, and the things they're asking for make a lot of sense. And so we do anticipate completing our regulatory work with Health Canada. They've even allowed us to do some work with clients up to this point. So they've been a much easier regulatory body to work with. But just it's very complex, because not every country, but a lot of countries have, excuse me, let's get rid of that. A lot of countries have their own they have their own agenda. They have their own health ministry. They have their own. And so anyway, everything's a little bit different.
I am so sorry to interrupt the flow of your presentation. Thank you for that. I think that's a great explanation, and I think that's going to help a lot of people when it comes to any type of newly implemented natural ingredient. It's going to really give them some reassurance that they're making the right decision in choosing this product. So I'm going to shut up and let you get back to your
And I'm just going to add to that real quick, Barry. I think that the majority of the issues that the EU and other regulatory bodies have is that there is potentially a possibility of maybe like a transmissible disease coming from a human cell, right? And so they don't want to have a product that potentially has some kind of disease in it. I just want to mention that the FDA has strict regulations on testing cells for organ transplants. So we actually follow the FDA regulations for organ transplantation before we even take these cells from human beings. And then we further test them again after their cells so that there's confirmed there's no transmissible anything that's coming off of these human cells. But I think in general, that's usually the concern, is because if you're, you know, I don't want to say a bad actor, but maybe you are just not following all the regulations properly, there's the potential that you could harm someone.
Yeah. And, you know, I don't want to scare anybody. And it's certainly, we do it right. We don't cut corners. Everything we do, we have, you know, pathologist reports on everything. But you can imagine if you're a regulatory body for an entire country and you just, or entire group of country, you just want to be careful. But I don't mind saying this on the record. I disagree with their philosophy because they're being extra pedantic about an issue that's really a non-issue. That, you know, that might have been an issue back in the 80s, but we have very, very good testing means now, and they just haven't evolved to where we are right now. That's my opinion, but you know, you guys got me on here to hear my opinion, so there you go. All right. So we're going to try to jump back in. Barry, I don't mind the question at all, but I'm a little derailed. I just got to get back on into the zone. One of the things I wanted to tell you guys about, which not all, this is where I start to sound kind of salesy. I don't mean to sound that way, but it's one of the reasons why we were slow to get into this in the first place, is that not every lab does it the right way. Not every company, you know, gets a Dr. Dave with his, you know, vast knowledge, or not everybody's trying to culture themselves in the right way. So for us, we are really transparent. We want everybody to know who we are and what we're working on. It was a must for us to do it the right way and give you guys and all of our customers a real product. And so in addition to culturing the stem cells in our labs, according to the best in class sort of standard operating procedures, we took it a step further. And this is something that Dr. Dave pioneered. And he's so modest. I wanted to take this slide so I can really brag about him, so you guys can love on him later. But this is a fascinating idea. And what he's really doing is he's demonstrating a deep knowledge of how the skin biology, how stem cell biology actually works. So your body is constantly responding in a dynamic way to the environment that it's in. If you go out in the sun, your skin cells immediately start reacting to that UV radiation, that sunshine. If you get, you know, punched in the face like I was this morning, your body immediately starts reacting to that trauma, you know, and so it's constantly dynamic. If you eat a burrito, your body starts a whole bunch of processes to make it. So Dave imagined a way to take advantage of that dynamic way that the body works. By us, the scientists, we put in stimuli in with the stem cells that make them react in a way that we can anticipate. Because if you know what it's going to do, you can start to plan around the products, the proteins, the growth factors, the cell signals that a stem cell expresses under that particular stimuli. So he ideated, and then we began to research this process of stimulating the stem cells while they're growing in a variety of different subtle ways, sometimes not so subtle ways, so that the stem cells have to react. And we wrote that up as a patent, and we calling it dynamic cultured media. So not every lab is even growing stem cells in what we would consider to be the appropriate way. That's the healthiest way. We take it a step further, and we try to, the best way I can simplify this for you is we try to make a stem cell conditioned media that's specific to the cause that we're trying to address. So very simply put, if you guys came to us and said, man, we would sure love it if you could make us a product to address hyperpigmentation or melasma, we would look at the stem cells and say, what are the factors and the proteins that actually address that? And how can we stimulate the stem cells along a pathway to get more of those factors into a stem cell product? And so it's a fascinating, again, it's just cutting edge. It's a new way of doing it. We do it. Nobody else that I'm aware of is doing anything like that. And so when you get a product from Factor 5, it's not a normal, I know that sounds sort of corny. It's not an everyday like as if stem cell condition media was an everyday product. But it's different from other laboratories because we've taken it a step further and we've dynamically tailored the stem cell condition media for the product that we're making. And that's something that we have a whole new, I'm not going to spoil it here, but there's another layer to that we're just starting to investigate here internally. But it's a fascinating field of science. I hope you guys get it, the idea behind it. It just takes advantage of the fact that your body is always reacting to its environment. And we are tailoring that to make really, really cool products for you. So, and Dr. Dave, he's the one who dreamt it up. And so I knew he wouldn't brag about it, so I had to do it for him.
Thanks, John.
How do I make it go? Okay, Dave.
John and Dave, do you want me to just share my screen instead?
Yeah,
Sure, why
maybe
don't you
that'd
do
be
that?
great. We feel like doofuses over here with nothing. Whose slide is this? You're mine, yeah.
All right, this is much better with video. So, so as John was mentioning, what we do is we take that dynamic cultured media and in essence, really what we're doing is we're tricking the cells. So we're growing these cells in the laboratory, but we're tricking it into believing that it's inside of the body and that it's responding to a certain type of trauma to basically heal and rejuvenate the skin. And so what we do is we take those proteins and our process now is that we flash freeze the protein to save it for later. This DCM, it contains over 700 different proteins that we've identified so far. It also contains exosomes. And then what we do is we add copper peptides and stabilizing agents to it to basically preserve those exosomes and to preserve the proteins that are contained within. And then we do a really cool process, and that's what you're watching here. And this process is called lyobead production. And so what you can see are these little drips being dripped into liquid nitrogen from this robotic arm. And those drips are basically the dynamic cultured media. They're hitting the liquid nitrogen and they freeze into these little beads, which are kind of shown up at the left-hand side, those white beads. I like to call this Dippin' Dot technology. Probably all of you have seen Dippin' Dots. Ice cream, this is the same concept, right? They just take the ice cream and they drop it really fast, do a real quick freeze in liquid nitrogen. But then what we do is we make it astronaut Dippin' Dot ice cream. We take that Dippin' Dot and we actually lyophilize it. And that's just a fancy word for freeze drying. And after that, basically, and I should mention that we do all this in these ISO class 5 sterile conditions. So basically under clean room conditions, so everything's sterile. And then what we do is we package those into these little vials. We'll show you a couple of images of those later. And then we also, what comes along with that is hyaluronic acid. And that hyaluronic acid is a sterile version of hyaluronic acid. And it's also injection grade hyaluronic acid, over 2,500 kilodaltons. And that is also packaged under sterile conditions. So basically it's a sterile product on both sides. You take that hyaluronic acid and you add it to those little astronaut Dippin' Dots that we've made. And they resuspend.
You're getting ahead of us. Don't do that. Can you advance the slide?
Yes.
He's killing the
Spoiler alert.
Spoiling it. It's the best in.
But so the key ingredients that are in this formulation, as I mentioned before, are the exosomes and the human stem cell growth factors. These are really important for rejuvenation of the skin, contribute to overall skin health. And then the hyaluronic acid, we all know that's an important component of the extracellular matrix that's in your normal skin, but it also has this awesome quality of being a humectant and binding up tons of water. Helps hydrate, soothe, increase the skin's elasticity. And then we also put in these copper peptides. And I think people don't really appreciate how important the copper peptides are to this. For ours, it gives the beads a nice blue color, but it also does something way more important. So GHK, that's the peptide, and it is attached to a copper molecule. And this is a clinically proven peptide that was actually identified in human plasma. And it's really, really important for hair growth, for rejuvenation of the skin after wounding. It boosts collagen production, it enhances elasticity, and really, really has been clinically proven to support skin repair after trauma.
I'm sorry. I just trying to
And now,
advance the slide. Oh, this is
this is the thing I spoiled before.
Yeah,
All right,
okay. So let me do this because this is my favorite thing in the world. Okay, there's a lot I really want to talk to you guys about this, but I'll try to be brief. Remember how I told you about how exosomes are very temperature sensitive? One of the things that we thought about when we're making our product is how do we keep the exosomes safe? So Dave's manufacturing process, he just showed you where we're dripping those drips. That's that dynamic cultured media that I was telling you about. And when it drips in there, it flash freezes. So that's exactly what you want. An exosome likes to be cold like that, flash frozen. And then Dave talked about adding stabilizing agents to it. Through millions and millions of different studies, what we realized was that you can actually freeze dry exosomes and they will stay wonderful, they'll stay functional, they'll stay biologically available, but you can't just do it on its own. You have to add these stabilizing agents. It took a long time and a lot of research to figure that out. But these stabilizing agents make that possible. So what that means is all the goodness that we figured out in our DCM gets to you by way of these lyo beads, right? These Dippin' Dot technology they're talking about. And then because it's freeze dried, when you add sterile hyaluronic acid, which we also provide in the kit, you can watch, and what you guys are watching right now, this is why I love this image so much. What you're watching is hyaluronic acid becoming infused instantly with billions of exosomes, growth factors, proteins, and copper peptides. So it's like you're watching you're watching the greatest ingredient in all of aesthetic medicine sort of being created in front of you. And one of the things I'm hoping that Tiffany will talk about in a minute is what it's like to be the clinician showing your client that you do this right in front of them. You actually add the HA. As I told you guys, we're in our manufacturing plant, so we got a shipment coming and Dr. Dave's running over to handle that. But you mix this in front of your clients, and I'd love for Tiffany to give us sort of a real world insider what that's like to do. Thank you for advancing it. So this is what our kit looks like. You kind of know what's in there now. You see the one vial is the sterile hyaluronic, the other vial is the lyo bead technology. You can see the beads come in multiple colors. The colors are really just a function of the ingredients that we put into it, right? The copper peptides do have like an interesting bluish color, and sometimes it comes out looking a little bit purple, which you see in your screen there. But that is what Exofecta is. It is the growth factors from human stem cells, all the signals that you get from stem cells, the exosomes, and it's from dynamic cultured media. And then you resuspend it with a sterile hyaluronic acid. The reason we made it sterile is because we wanted you, the clinicians, to be able to feel safe and comfortable about putting it onto somebody whose skin had been kind of recently abraded or wounded in some kind of way, without feeling like you're maybe not doing right by your client. And I want to just point out, as the guy who writes the checks around here, that was extremely expensive and difficult for us to do, is to make it all sterile. But again, we don't like to cut corners. We wanted to make it a really good product for you. So the HA is sterile. And again, I can't wait to hear what Tiffany's take on that is about what it feels like to mix this in front of the client.
And I also want to kind of piggyback on this too, because we've been talking about stabilizing agents, and I think sometimes people hear that and they're like, "Oh, I don't like that term." this stabilizing agent, what I want you to think about is the reason that it's really hard to freeze dry things and keep them biologically active when you rehydrate them is because think about what we're talking about here. We're talking about an exosome. So it's a package, and that package is wrapped in this lipid bilayer. So what happens when you freeze that is ice crystals form in the lipid bilayer, okay? And when you thaw that, the ice crystals basically rip apart the lipid bilayer. So what we're doing is we're actually adding something that basically keeps the lipid bilayer very soft and flexible and pliable, okay? So that those ice crystals never form and those ice crystals never rip things apart when they thaw. So that's the whole technology behind this. And, you know, we can go into that. I'm sure people will have tons of questions because there's been a lot of discussion over whether lyophilization works as a preservation method for proteins and things like that. But I can show you scores and scores and scores of literature that shows that lyophilization does work. And if you don't believe me, it doesn't matter because we've already validated that it works. And one of the places that we do this with is this independent research facility called UC Davis. So University of California, Davis. It's about an hour and a half away from us. And what we've done is we've actually partnered with them. They have a specific extracellular vesicle core facility where all they do is basically validate EVs and exosomes for internal researchers at UC Davis that work with stem cells, and also for external researchers like us, but also pharmaceutical companies, right? As we mentioned before, people are trying to use exosomes as drug delivery devices. People are trying to figure out exosomes as treatment for Parkinson's disease. Lots of different things that people are doing, and they need to be validated. And there's very specific ways to validate these exosomes. And we thought it is probably not appropriate for us as our own company to tell you, we have made these and we certify that they are quality. I don't think that's probably the best way to do it. We think going to a third party and having them tell you that they actually work is much more effective. And so
we're getting the music.
Oh, sorry.
Wind it up.
So anyway, we can give you information on this, but basically these can be validated through a number of different ways.
You guys, I want to just let you know, we know we could have a 3-hour exosome conversation with all of you. The Q&A is packed. We have so many things that we know we want to get to. So we're definitely going to have Tiffany. I want you to take your time. I want you to go through your portion of the presentation. And if we have a few minutes at the end to pick up a few of the Q&As, we absolutely will.
Certainly.
Okay. So I'm Tiffany. I'm an in-house esthetician for FACTORFIVE, and I had the awesome opportunity to work with Exofecta before it launched. And to tell you the results were amazing and mind-blowing. So this is a picture of a before and after of my client. You can see in the before, she has some ruddiness, some erythema, skin laxity in the chin shadow region, and it looks like smoker line. She's not a smoker, just kind of like a dull appearance. So what we did for her was I took it really simple. I was just like, okay, I'm only going to use this and let's see what happens. So I applied it topically, and I'll get into the modalities. I've seen that question. I'll get into that. But I just applied this topically for her, and you can see like the visible firming of her skin. The redness is diminished. And you can look at the dehydration lines under her eyes. Those are softened. You can tell she looks more supple, more hydrated. Her lips, the only thing on her lips is just the Exofecta. So I want to point out that this is good for all skin types, all Fitzpatrick's, including your sensitized and compromised skin clients. So sometimes, and I'm sure you have this like, oh my gosh, I want a chemical peel, I want to burn my face off, I want to, all the things, and they have either grade 3 acne, or they have a compromised barrier, or it's a first-time client. That is not going to be your go-to treatment. So having Exofecta in your arsenal of supplies for any of those patients is really just going to, increase the trust and loyalty in your client, which is going to give you client retention and also increase your revenue. So one other thing. Yep, that's good. Okay, so how do I incorporate it? You can go to the next one. A plethora of modalities. So One of my favorite stack treatments is the Factor 5 Renew Dermal Solution. If you don't know about that, that's a whole nother webinar. Reach out to us, reach out to me, and we'll give you all the details on that. It's amazing. So you can use it with chemical peels. I absolutely love to use this as my glide for microneedling, nano infusion, after dermaplaning, after microdermabrasion. You can use it after any kind of injectables that you have. Sculptra, it heals nicely, helps with bruising. And there was ultrasound. Oh, and cold jet plasma. I have found Barry is a testament to this. You open up those channels to the skin and you apply this product and it's going as deep as an injection layer. So you're going to be blown away by these results. You can go to the next one. So this is just again her before and after. Honestly, the first time I used this, I was really blown away. I thought I was hallucinating. I could see that I worked on the left side of her face, and I was like, is her face more firm than the right side? So I asked her to smile, and actually, her smile was crooked. That's how firm, an instant that the results were. So. This one, Sweet Sammy, she does a lot of microneedling, micro channeling. This has really reduced her downtime for her clients, which is what our clients want. They want the results, but they don't want the downtime, and this allows you to give them that. The last one, Janelle from California, she's an amazing esthetician. She used this, she applied it topically, and she used LED with it. So, I mean, you can look at her results, they speak for themselves. Everything, they just appear more firm, youthful appearance. And one thing I wanted to point out about a compromised skin or someone that comes in with their goals, most of the time, if they do have a condition, they still want this anti-aging or age management goal. And this product allows you to do both. It allows you to help treat conditions as well as help with skin laxity, firmness, and just overall youthfulness and health appearance to the skin.
Can I ask you a question, Tiffany?
Sure, of course.
So this is one of the things that I was trying to express, but I really would like to get your take on it. So you're in the treatment room, and this is a visual medium, right? Like everybody likes, that's part of the reason you guys set up the soft lighting and the comfortable bed, and you just want it to be a comfortable environment. What's it like to mix Exofecta with in the room with them watching?
I did this morning on Sweet Tracy, and I was like, Tracy, watch, you got to watch this technology. And so they're always blown away by it. And that they honestly, they just asked for EXO. That's like this morning when she came in, she's like, I'm all red, I'm inflamed. Like, can we just do some EXO? So watching it and feeling it is just really goal changing for your clients.
I have to say, when you're doing the level of skincare treatments that this client is expecting, if you're doing jet plasma, if you're doing needling, if you're doing these advanced treatments, your treatment room is a little bit more bright.
Yeah.
And so they can see what's happening. And I think that the more technology and the more sophistication you're exhibiting in every step of the treatment adds to the credibility of the treatment. And I love the way it looks. I love how you have to mix it. I loved watching you do it in Las Vegas when you did it on me. We combined it with jet plasma in Las Vegas, and we did a whole post about it, and it got a lot of attention. Anytime we post about this, we get a lot of attention on it. So I have one question. Do you think that Exofecta is more effective when it is applied to treatments that create channels in the skin versus just like a topical chemical peel?
I mean,
I like to do it all together.
You do?
I do. And it depends on the
Okay.
client, their budget, what are they doing after? Where are they going after? With,
Okay.
RDS, it's a whole different game.
Okay. I was just curious. I didn't know if you thought that, let's say, for example, you had a client that didn't want needling. They just wanted a refresher peel. And so they're just doing the peel and then putting the exosomes on, which are pretty much at that point, they're not getting into a channel. So do you think that they're more effective when channeled into the skin, like with a needle or a microchanneling device or a nano infusion device?
You know, honestly, I've done it so many times that I do see the same result. I think if you're already using an infusion of some sort, use the EXO. But like, I don't think I'm not going to get a result if I don't use an infusion. That's not the case. Like, I have
Okay,
before and after, before and after of where just applying it gives an incredible result.
love that.
That
Thanks, Tiff. I
segues
appreciate that.
right into a question that I think John and Dave can answer. So in the Q&A, Jacelyn, she wrote, can you explain FDA regulation around applying exosomes after needling or to broken skin? I hear that it's not legal to perform that way. So do you have any feedback on that?
Yeah.
And Jenny, I would also hop on and say, is the person that asked that question in the United States or are they in Canada? Because I think when she put it in chat, she used the word here, and so I don't know if she's Canadian or American.
Well, if she's asking about the FDA, that's under the purview of the United States of America.
Oh, she did say FDA. I'm sorry.
So the answer is the exosomes are not approved for medical claims. Okay, and again, I don't want to sound like I'm trying to oversimplify this, but what we're really talking about is what you're saying to people and what kind of claims you're making. And the reason this distinction had to be made by anybody is just that people were out there claiming that they're going to inject exosomes in your spine and heal you from whatever condition. They're making very, very wild, very over-the-top claims. Now that might be the future of exosomes, but that's not where we are right now. And so the FDA is very strict about what you're telling people. They don't want to misinform the patients of the world. So if you're telling them, I'm going to put this exosome product after your microneedling, and it's going to cure your erectile dysfunction, that's a medical claim and you shouldn't say it. But if you're saying, hey, I'm going to put this on there, it's going to help you have brighter skin, it's going to help you, you're going to heal faster, and you're going to have more luminous skin, and you're making aesthetic claims, you're teaching the patient correctly, then that is okay according to the FDA. What they're concerned about is you misinforming them about the benefits of the product or making a medical claim that hasn't been appropriately validated with clinical testing. Does that make sense?
Makes perfect sense.
So I know it's a little nuanced because we always think of the FDA as sort of validating whether the product works. But what they're really one of the things that they're doing is just trying to prevent misinformation or too much craziness in the marketplace. You and I, or all of us here in the aesthetic world, we sort of know what they're talking about because it's less regulated here. So companies are saying all kinds of things, and it is confusing. The FDA is trying to prevent that confusion from getting into areas where the people are talking about life and death types of treatments, cancers and different related diseases, heart disease and whatnot. So they're very careful about regulating what people are telling their clients about products like Exofecta.
Perfect. Okay, so if you're ready to bring it home.
Ready?
Is it me? Yeah.
Oh, go ahead.
Sorry. Thank you, lovely Tiffany Underwood. We are so grateful that she's, it's great to have a clinician on here with us. So the EXO, this is the part where I'm very, very, again, I'm very passionate about this. I have gotten to know some of you, my customers, our customers out there, and I appreciate you very much. We take it to the next level in the science, but we try to keep the price where it's approachable for everybody. So this is our kit. The total cost is $400. Because of the way that we do the lyo beads and freeze them like that, you can store it at room temperature in your practice. You don't need to store this under any special conditions. You can leave it right on the shelf. Our hope is that every esthetician at whatever level you are, whether you're just starting out or whether you've been in practice for 30 years, that you can afford to get into a box of Exofecta for that bougie client that you have that wants that next level thing, and that you can use it as needed. So there's 5 treatments in a kit, and the kit is $400. So it comes in about $80 a treatment. And we have across the country, we've had a lot of feedback from different estheticians telling us, in this area, I can charge this much for an exosome add-on. In this area, I can charge that much for an exosome add-on. But we took your economics into our thinking and our rationale when we priced this product, so that you guys could afford to get a box and hold on to it sufficiently long to make your money off of it, which is one of the things we hope you can do. And the last thing I'll say on that is, since our inception, when we began our company in 2015, we have always had, and I still maintain, I am the founder, I am the CEO, the buck stops here. I have a CEO approved money back guarantee on all of our products, pretty much no questions asked. Again, I do that out of respect for you and your business. I don't want you to invest in something that you don't end up liking. And so what I try to tell folks that are concerned about the price is that your money is safe here, because if you don't like the product, I'll just give your money back. So it's not really, you're not taking a major economic cash risk. It's just a risk of whether or not this product works for you in your practice. But, so it's not a major cash risk. It's $400. If you don't like it or you don't feel like it's working, just say so and we'll return your money.
I want to also mention that we have done studies that show that after you resuspend those, the lyo beads, and they're in the liquid form, you could put that in the refrigerator and those will keep for about 2 weeks. So we know the protein concentration and the exosomes are still there. They're stabilized. So if you, like, for whatever reason, didn't want to use a whole vial on someone, you could potentially use that for other treatments.
Let's see. What am I supposed to tell folks? You can go onto our website and apply to become a customer. And that's the web address right there, wholesale.factor5skin.com. And applying is not that big a deal. You just click on there, you fill out the form, and it asks for your esthetician license, things like that. But that's it. That's how you do it. And now we want to take any and all of your questions.
So the Q&A is open. We are at 3:21. So we can download the questions, the Q&A, and we can send it over to you. I'm more than happy to stay on to answer questions. We still have almost everyone who joined. So if you guys are open to staying for the Q&A, we can absolutely stay. Let me know in the chat, you guys, if you're able to stay for Q&A or if you want us to close up shop. No
Yeah,
one's
people
comments.
say they were hopping off, but you guys, just as a reminder, this is all going to be recorded, and as long as you registered for summer school, you'll get the replay in your inbox in 24 hours. So I know you guys are going to need to hop back on and rewatch, because some of this you want to reabsorb a little bit more.
Okay, and I'm sorry to cut you off, Jenny, but there's question after question coming in the chat. Don't put it in the chat, because if we can't get to it, it's gone. You have to put it in the Q&A. So we'll stay. We've got a lot of people saying they're going to stay. So John, the first question you can see is from Alicia. Do you see it there on your screen if you pull up Q&A?
Are harvested or collected, I guess. Are they all kept together regardless of their contents, or are they separated into groups based on what they contain? Great question. The answer is, right now, according to the science, all the science I know, we don't know how to separate them by group. Right? They're that small and they're that complex, and there are some science that can determine what's in them. But there is no thing. I mean, I don't know if we, as a body, if we really appreciate just how small these are. Like, Barry, I'm not trying to correct you or whatever, but these are so small that if somebody was doing a gentle peel, it would still just go right in there. You don't need to do
please correct me.
Yeah.
That's the question is, correct me, because if I have the question, there's other people who have the question too, and that's what we're here for. I love it.
Yeah, you don't need a microchannel. There are some of our growth factors probably are majorly enhanced by microchanneling, so that is a real thing. You're not off base there. But especially with Exofecta, they're so small, and a nice, even a gentle peel is actually opening up apertures for our growth factors and exosomes to get into. It's just doing it at a very, very small level. And so it's a wonderful application for our tech, because it doesn't lead to a whole lot of downtime, but the good stuff still gets in.
We are doing studies to figure out the different multitudes of things that are inside of these EVs and be able to separate them out. But currently there is not technology to actually do that, to say like, this is the RNA pool and this is the protein pool and all of that. That's coming.
So,
And then,
sorry.
I'm sorry, go ahead. Were you done with Alicia's question?
Well, she had a second one that I was going to take a stab at.
Oh, here, I can go back. I thought, I didn't realize it was a second question. Do you see it there? When EVs are harvested or collected, are they all kept together regardless of their contents?
That's the first one. The second one was,
If they're separated into groups, then is it known what benefits are of each group?
I don't think I know the answer to that. Sorry. Yeah, we're getting a little, we're bouncing around between screens trying to figure out which question we're answering.
Okay, so well, you're going to get a download of this, so you'll be able to
Okay.
go back and answer it. So then we have Mary Ellen. Can these products be used on oncology patients with compromised skin?
This is okay. So scientifically, there's absolutely no reason why not. But whenever we get into something like an oncology patient, we don't want to be the ones that put you into an awkward situation with your. So we always advise that you do your treatments in harmony with their provider, their physician who's treating this patient. So there's no reason why not. In fact, I don't want to speak for Tiffany Underwood, but she's sort of a local expert on this in her own way as an oncology patient. There's no reason why not, but we do not advocate for you to do this sort of in isolation of the treatment paradigm, the treatment protocol. Oncology is a very serious thing. We want you to do everything you do in a harmonized way with their providing physician.
Okay. Megan asked about opening order. Caitlin's answering that. Do you want to just give everybody an idea of what the opening order is?
Opening order requirement, you can order 1 box of Exofecta.
Okay. And then Tanya Riley asked, I've heard you can remove a number of beads and customize the amount of exosomes. Is this true?
This is such an awesome question. I mean, Tanya, you're going straight to this is a fascinating question because it gets right to what Dave has done and what we have done by making this into beads is if you understand what you're talking about the number of extracellular vesicles, you can start to do a little bit of math and try to figure out how many EVs, how many exosomes are in each bead. And you can begin to try to separate it out and there's a whole fascinating new world in there baked into that question. It's not something I'm prepared to dive into too deeply at this moment. But Tanya, I love your instincts. And I think you're going right where we were imagining the future as well.
Perfect. And then Tiffany asked, do you have a protocol for the scalp with Exofecta?
Should we go to our Tiffany for that one?
Yes, we have protocols for jet plasma if that is what you're going to use as your modality or channeling.
Perfect. Thank you. And then Lisa asks, are there contraindications and can you use beyond the orbital bone and lid?
I always do.
Amazing. Awesome. Well, we did it, you guys. We answered all the Q&A, and now the Q&A will be in the recording that will go out to everyone tomorrow. So that's great. And it'll also be in the recording that lives on the app. So we did it. And I am just, first of all, I don't think we've ever had a webinar that's gone an hour and a half. Like, I don't.
And could go longer. If we
Another hour.
had 2
I'm
hours.
sorry.
No. But the best part is it's kind of like that like a war like the most amazing movie. You walk out, it was 2 hours, and you never once looked at your watch to see if it was over. Like, I only looked at my watch because my phone is laying here, and I'm like, wow, we're at 3:06, and Tiffany hasn't even talked yet. Let me see where we're going. This was amazing, you guys. I love working with you.
Well, thank you very much. It's very, very generous of you. Yeah, we have a tendency to go long. This is our favorite subject. But thank you for your attention, everybody. Thank you for having us. It's just really gratifying to have a forum to talk about our work.
It's amazing. Jen, any final thoughts from you?
I think this is the perfect example to the entire Factor 5 team that we need to do a follow-up. So just tuck that in the back of your head, Caitlin and John and Dave and Tiffany. We would welcome you guys back anytime.
Awesome.
Absolutely, absolutely. And August is Science Month. So this is Dr. Dave and Dr. John's invitation to write up something on exosomes and really talk about how the EU has certain specifics and the FDA has specifics, and you work within the FDA, not the EU. So I think that would be a great topic to discuss the science of exosomes. Tiff, any final thoughts from you, my love?
No, I mean, you can't go wrong. You need this in your arsenal. Every practitioner needs this, truly.
Love.
And we're
love.
here to support you with anything you need and how to implement it. We got you.
Amazing. Well, you guys, thank you so much for being panelists today and taking time out of your schedule. We truly appreciate that. To the unbelievably engaged attendees who were keeping us on our toes the entire time with chat and Q&A and just question after question after question. You guys bring the energy, you bring the fun. It really makes this way more fun for us to talk to an engaged audience versus just like giving information out into the interwebverse. So it's much more fun for us. So personally, we just want to say thank you for that. If you're one of the more than 1,500 people who have registered for summer school and know that you're going to get this in your inbox tomorrow, you guys rock. And we've beat our summer school registration for 2025. We only had about 1,400 last year, so we're doing really, really well. Summer school is always well received, and we're so grateful for you guys for that. But if you're one of the more than 8,000 people who have the Lipgloss and Aftershave mobile app on your mobile device and know that you can get your education 24 hours a day, 7 days a week from your mobile device, your tablet, whatever type of mobile option you have that has the app on it, you guys know that it is the best source for esthetician education. So you rock. If you're on the app right now, keep scrolling, keep learning. Have a wonderful day, everyone. Bye-bye.
Guys.